Good afternoon. Today we're going to discuss Chapter 6, How the Cells Harvest Chemical Energy. So we've been building and building on this idea for a couple of weeks. We started with biological molecules, the cell, and most recently we talked about the working cell. And sort of got an idea of the enzymes and the ATP and all of the other things that are necessary for a cell to do any sort of work. So with chapter 6, what we're going to do is talk about energy conversion. We're going to talk about how cells need energy to function and how those cells get that energy. So what you can see here in this picture is a picture of a giraffe. And the giraffe is obviously eating some leaves. And basically what that means is that the giraffe is going to obtain energy from those leaves in the form of sugar. And then the body is going to convert that into ATP for cellular work. Where are we? Here we go. So basically, cells need energy no matter what type of cell that it is. And there are many ways that cells can obtain energy for their functions. Most of them have something called an electron chain to pass the energy along. But basically, there are 3 different ways that cells can obtain energy. One is called aerobic respiration. And aerobic respiration is the use of sugar energy using oxygen. And basically, what we're going to talk about here, and what you see in this picture, where oxygen is used to produce water, but that is sort of the end product. The real star of the show is creating ATP from glucose. So there's aerobic respiration. There's also fermentation, which is the partial breakdown of sugar without oxygen. And the final electron acceptor is some sort of organic compound. In our case, we're going to talk about something called pyruvate. Then there's anaerobic respiration, which is when the final electron acceptor is an inorganic compound. We're not going to do anything with anaerobic respiration in this class, but you will, if you take microbiology, talk about anaerobic bacteria and organisms like that. So chapter 9 is really going to focus on aerobic respiration, and we'll do a little bit with fermentation at the end. The process of obtaining energy from food is going to be a catabolic pathway, right? We're going to release energy to break down molecules. Remember that our overall metabolism is the sum total of the energy creating and the energy producing. Energy creating and energy producing reactions in the body. And when we focus solely on the breakdown of organic molecules, then that's going to be a catabolic process. So basically what we're going to talk about is the use and breakdown of glucose for energy. Now, the body requires ATP energy. Glucose is a giant molecule compared to ATP. So we have to break down that glucose into small bits of ATP for work. Basically, we're going to have a stepwise transfer of electrons and stored energy, which is something we're going to have to sort of pick apart piece by piece as we go through this process. And this is a very process-heavy, very complicated set of topics that we're going to now talk about. So you may definitely want to consult some of the other extra videos and things as you start to learn about cellular respiration. So basically, cellular respiration is going to provide the ATP for cellular work. Cellular respiration is going to be the right-hand side or the bottom part of this graphic. The top part is photosynthesis. So what this graphic is showing is that the light energy comes from the sun and then it hits the plants or anything with a chloroplast. The plants then convert that light into organic molecules and oxygen. And then those organic molecules can be broken down by the mitochondria into ATP and a little bit of heat. The byproducts, carbon dioxide and water of cellular respiration, are then rearranged using light energy, and the cycle goes around and around and around and around. So it's very important to understand that this cycle of cellular respiration and photosynthesis is basically to um, sort of watch the flow of the sun's energy through the systems, right, to ATP and then heat, right? So if you see the light energy goes into the chloroplast, then it's broken down by the mitochondria, and then it powers cellular work through ATP. So let's think about what we're talking about here. We're talking about glucose. And glucose, if we think about it in terms of dollars and cents, right? Sugar or a glucose molecule is going to be like $100 bill, but cells only take nickel and dimes. So we need to break down that $100 bill into nickel and dimes for the body to be able to use it. So our body requires a constant supply of energy from the food we eat and the oxygen we breathe in. The conversion of glucose into ATP is only about 40% efficient, right? The rest is lost as heat. Now, the activities of the cells require ATP. The average person needs about 2,200 calories of energy or calories of food per day. And those calories are a direct representation of how much energy the body needs. So we have these two reactions, photosynthesis and cellular respiration, that we're going to go into in detail. And we're going to start with the cellular respiration. So here we have a woman running, and she's breathing in and exchanging oxygen and carbon dioxide. She is, that oxygen and carbon dioxide are transported in the bloodstream to the muscle cells. And when the oxygen comes in, it goes to the cells. It helps to process the glucose. The resulting molecules are carbon dioxide, water, and ATP that are then sent back into the bloodstream and used or excreted. So you'll need to know this calculation at the bottom, or this chemical equation at the bottom. Glucose plus oxygen yields carbon dioxide, oxygen, and ATP, and also heat. This is a better way to represent it. One glucose molecule is reacts with 6 oxygen molecules to give rise to 6 carbon dioxide molecules, 6 water, ATP, and heat. Number of ATPs is somewhere between 32 and 36 ATPs are given off in this equation, plus heat. So if you can look at this equation, you'll see that glucose is C6H12O6, right? You'll also see that oxygen is O2. And if you look at those two molecules and see how we can reconfigure them, right? If you take the oxygen and the carbon, the oxygen molecule plus the carbon from glucose, that forms CO2, and then the H2O is left over from the glucose, forms the water. So you can see how those get rearranged in the chemical equation. So what we want to do is talk about systematically how we're going to take the hydrogen from the glucose and bond it to the oxygen, right? That's what we want to do here. We're going to take the hydrogen and bond it to the oxygen, and then we're going to end up with carbon dioxide, water, ATP, and heat. So the movement of the hydrogens is going to represent the release of energy. And we're going to go through that piece by piece. Now, the ATP production video here, the overview of cellular respiration, you really need to go find that in Mastering and take a look at it. It really gives you an excellent idea. It's an animation on this whole process. So I would go watch that first, and then watch it again after the PowerPoint. So when we talk about using cellular respiration to generate ATP, we're going to have to talk about a whole bunch of different types of molecules that we haven't talked about before. One of them is going to be something called NAD+. You can see that on the left-hand side in the little gray square. It looks like a nucleotide, which it is, and it is an NAD+, so it's an ion. And what it's going to do is it's going to take some hydrogens through, see the purple rectangle? Through that purple rectangle enzyme called dehydrogenase. And remember what we talked about just two slides ago was we need to figure out how we're going to get that hydrogen onto that oxygen to make water. So we need to dehydrogen that glucose. So the enzyme that's called dehydrogenase, removing hydrogen, is the perfect name. So it's going to take a couple electrons and a couple hydrogens from that glucose, and it's going to stick it onto something called NAD+. right? And so that's important. NAD is a coenzyme, and dehydrogenase is an enzyme that we need to um, make sure we understand how it works and make sure that we know its name when we come upon it. So to create ATP, the cell has two ways it can do that. One way it can do that is called substrate level phosphorylation. And remember when we were back in chapter 5, we talked about phosphorylating ADP to create ATP. And so what we want to do is add a phosphate group to that molecule. That's called to phosphorylate it. And so we want to, if we look at the picture on the left, we have an enzyme, and it's going to take a substrate that has a phosphate group on it, and it's going to bind that phosphate group to the ADP to make ATP. So that's a very simple way, an enzyme transfer of a phosphate group from an organic molecule to ADP. This occurs in organisms that don't have a mitochondria. So this is a primitive or more earlier form of ATP generation. Almost any organism can do this. Bacteria can do it, plants, animals, fungi, protists, everybody, every organism practically can create ATP using substrate level phosphorylation. This is an earlier version. As organisms became more complex, they needed more energy. When you need more energy, right, you need to have a way to be more efficient. And that's when we end up with something that looks like this crazy picture. This crazy picture is called oxidative phosphorylation. You can see that at the bottom. So I'm going to kind of walk you through it. So obviously in the middle of the page, there's a membrane. Right, now this membrane is actually on the inside of a mitochondria. A mitochondria is a membranous organelle. So this is actually on the inside of the mitochondria, and we have a series of membrane proteins, 1, 2, 3, 4, Q and cytochrome C. Each of those membrane proteins have a role. On the far right, we have an enzyme membrane protein called ATP synthase. To synthesize means to create. And so if we're creating ATP, right, that's an enzyme, that's a membrane protein that creates ATP. So we have this whole circus of things. Now, on the left-hand side, we have those NADH molecules. We're carrying the electrons from the sugar, right? You'll notice that all of the hydrogens go up into the orange. They get pumped out into what's called the intermembrane space. Now, what you'll notice is that on the top of the intermembrane space, we have a lot of hydrogen, right? That's a high concentration of hydrogen. In that golden yellow area on the bottom, low concentration of hydrogen. So if you follow the dotted lines, you'll see that those hydrogens they create a gradient. They want to pass down through that ATP synthase to achieve or try to achieve equilibrium, right? And so that goes back to our diffusion and membrane transport discussion from how cells work. So we're talking enzymes, we're talking membrane transport, but basically the cells are going to use ATP synthase to take advantage of the concentration gradient formed by the hydrogen ions, right, up at the top, as a result of the electron transport chain. So some of the proteins are pumping the hydrogen ions out. There are more hydrogen ions on the outside, and using the potential energy of that movement, ATP synthase creates ATP. So there's going to be a downward flow of hydrogen ions through that ATP synthase molecule, and that's going to create ATP, which you see at the far right on the bottom, that little starburst ATP. So we have this whole series of membrane proteins that are contributing to moving those hydrogens along, moving those electrons along, separating them out and creating this gradient. And as they flow down through ATP synthase, we make ATP. Once there, you'll notice in the middle of the page, you see oxygen. Now, oxygen is there to grab up and gobble up all of the hydrogen that comes down through. So this is a never-ending process. This process continues all the time, which is good because we need energy all the time. As long as we're breathing, right, we have oxygen and we have food, right, so we can get some electrons, we can create energy. The oxygen inside of the membrane is always going to scoop up that hydrogen, so that's always going to be a gradient. This is really difficult to comprehend, so make sure you're looking at some videos, you're looking at and reading about the oxidative phosphorylation step. All right, so both of those steps are actually the end, all right, is where we want to create some ATP. So if you look at this picture, this is the overall process of cellular respiration. It's 4 stages long. You'll notice at the bottom, the blue rectangle called glycolysis uses substrate level phosphorylation to create ATP, that enzyme driven. The citric acid cycle does as well. On the far right, we have oxidative phosphorylation, which is that membrane driven, gradient driven, high output, high energy producing process. So there's all kinds of steps that go from the very left where you see glucose all the way through to the end for ATP. And so we're going to kind of step through things piece by piece, see if we can sort of understand what's happening at each stage. One thing I do want you to remember and notice at the top of the page is that you have a animal cell on the left that has mitochondria. You have a plant cell on the right that has mitochondria. Animals, plants, fungi, and protists all have mitochondria. So remember that any eukaryotic organism can take advantage of mitochondrial creation of ATP. All right, so let's go through each step. So this is glycolysis. Glycolysis means the splitting of sugar. It's an exergonic reaction, meaning energy releasing. We're creating a small amount of ATP using that substrate level phosphorylation step, that enzyme driven step. And basically what happens is we invest a couple of ATP to sort of get the process started. However, if we invest 2, we get back 4. So there's a net gain of 2 ATPs. Ooh, excuse me. We also end up with 2 pyruvate molecules and 2 what are called NADHs. Now remember, those NADH molecules are going to be what's transferring those hydrogens over to the last phase of oxidative phosphorylation. When we take the glucose molecule, which is a 6-carbon molecule, we will create 2 3-carbon pyruvates. So basically taking glucose, cutting it in half, we're creating a little ATP and a little NADH. There are 9 chemical steps in 2 phases. So there's a lot going on. You don't need to know about this, but just sort of have an idea that we are adding glucose, we are splitting it in half, and we are creating at the bottom 2 molecules of what is called pyruvate. Right? So those pyruvate molecules go on to the next phase, which is the pyruvate conversion, where we have 3-carbon pyruvate, gets further broken down. We take off a couple more hydrogens, because remember what this pyruvate is. It started out as C6H12O6, right? Now it's 3 carbons, but there's still associated oxygen and associated hydrogen. The hydrogen is being picked up at the top by NAD+. The oxygen and carbon are being given off as carbon dioxide, and we end up with what's called acetyl coenzyme A. So in glycolysis, for each molecule of glucose that we oxidize into pyruvate, what are we getting? Right? Remember, so we put in 2 and we get out 4, and then we also get NADH and pyruvate. So what do we got? All right, so the answer here is going to be C. We have 2 molecules of ATP are used in the beginning, and 4 molecules of ATP are produced for a net gain of 2. All right, so we have some ATP that's produced, very small volume though. In the pyruvate conversion, there is no ATP created. We're just creating more hydrogen ions to be used at the end. All right, so the pyruvate conversion is prior to the citric acid cycle. The pyruvate is modified inside the mitochondria to form acetyl-CoA, and carbon dioxide is released as a byproduct. We also get some NADH produced as well. Then we end up at the citric acid cycle, which again, exergonic reaction, right? We're breaking this down, we're releasing energy. This happens inside what's called the mitochondrial matrix. There are 8 steps to this process. We are going to create some ATP, as you can see that at the bottom. We are going to get some NADH and another transport molecule called FADH2, right? And all of those brown rectangles with the little negative signs, which is showing electrons, are going to be shuttled over to the electron transport chain and the oxidative phosphorylation. So this is the citric acid cycle. Acetyl coenzyme A enters the cycle. It goes through citrate, alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate, and sort of cycles around. What we need to know, we're creating ATP, we're creating NADH. And at the end of this process, you'll notice that there are 2 carbon dioxides that leave. There are only 2 carbons that entered. So at this point, the glucose molecule is completely broken down. All that's left is the NAD pluses that we need to NADHs that we need to go figure out. And that's where we end up back at our oxidative phosphorylation step. Again, we're getting those electrons, those hydrogen ions from the other 2 cycles. It occurs across the inner mitochondrial membrane. And then we're going to produce upwards of 32 ATPs, right, in this process, which is exactly what we want. The hydrogen ions are bumped along, the electrons are bumped along, and everybody comes back together down at the bottom in the middle where we create water. The most important part of this to understand is that we took all of the hydrogens that from the glucose molecule, we created this gradient. The gradient then flows down through the ATP synthase molecule, creating ATP. It's like a faucet. The good thing is inside there is a oxygen waiting there to capture all of that hydrogen that comes through. So it never stops, never ends. So theoretically, if you add up all of these processes, you can have about 36 to 38 ATPs. So here are some interesting questions, right? For example, which stages give off carbon dioxide? Right, the pyruvate conversion and the citric acid cycle. This image here, figure 612, is the one that is the most important to understand and memorize. If you can understand this right here, you will have no problems on the quizzes or the exam. This shows you the reactant of each. It shows you what is produced by each. It shows you where the energy is created. It shows you where water is produced, where the electron carriers of NADH and FADH2 come into play. So this is really, excuse me, I don't know why I'm so tired. This is really where you can really put together an understanding of the cellular respiration. You have a picture of the mitochondria sort of behind the whole thing. So you'll notice that glycolysis happens outside the mitochondria, right? And then everything else happens within or across the mitochondrial membrane. So understanding this, very, very important. So what you would want to do is sort of give yourself a quiz. Where is oxygen used, right? Oxygen is used in the oxidative phosphorylation step. Which of these 4 stages create ATP, right? Glycolysis, citric acid, and oxidative phosphorylation. Which of these 4 stages produce carbon dioxide? The pyruvate conversion and the citric acid cycle. So kind of quiz yourself in that way, because those are the kind of questions that you're going to see on the exam. What happens where? All right, so this is an important one. All right, here's a question. What pathways generate these electron carriers? Right, so where do we get NADH and FADH2? Glycolysis creates NADH. The pyruvate creates NADH. And citric acid cycle creates NADH and FADH2. Right, so those are important to note. Right, so this question would be A, B, and C. Oxidative phosphorylation doesn't generate them, but it uses them up. What about this one? Newborn mammals have a specialized organ called brown fat, where cells burn fat to carbon dioxide without capturing the energy to reduce electron carriers. So cells burn fat, right, without creating ATP. So if we're burning fat and not creating ATP, what are they doing, right? They're generating heat. It makes sense because newborn baby mammals, right, very small, but they're warm-blooded, so they need heat. So if we're going to take the fat to carbon dioxide but not create any ATP, we're probably using it to generate heat. All right, so when we talk about, again, creating energy, right? What we just went over was aerobic respiration, the use of oxygen to power the mitochondria to create a lot of energy. Another way to create some energy is through the process of fermentation. Now, basically, fermentation is just glycolysis. This is a strange graphic because it starts at the bottom left with glycolysis, that that black 6-carbon chain. You follow it to the right, you create 2 pyruvates. In this case, notice how there's no oxygen, that little red hashtag, little red slash mark. That means there's no oxygen. So this little organism, if it wants to make energy, has to do so in a different way. And so what it's going to do, it's going to ferment. So those 2 pyruvate molecules are going to go up and to the left. Now, if we're in a yeast organism, it's going to create ethanol and carbon dioxide. If we're in muscles, like human muscles, we're going to create lactic acid. So fermentation is a glycolysis where the carbon dioxide is removed and oxygen and NADH is oxidized back into the system as lactate or ethanol. Right? So it's a partial breakdown of sugar for energy. It allows organisms that don't have a mitochondria to make use of sugar. Very important. Some organisms ferment alcoholically, right? That would be a yeast, where alcohol is the finishing product. In a human muscle, right, we would ferment and create lactate or lactic acid fermentation. And everybody's experienced that when you work out and your muscles have that burning sensation, right? You've created a byproduct of lactic acid in the muscles because of a lack of oxygen. Working out too hard, squeezing your muscles too much, right? Lactic acid. Now, some organisms like yeast are what are called facultative anaerobes, right? They can either ferment or they can respire, depending on if oxygen is in their environment. Our muscles can ferment or they can respire, but they can't ferment for a really long period of time because lactic acid is damaging. So the yeast, just switch it on and off. They'll either ferment or they will respire, depending on if oxygen is available in the environment. When we're looking at the process of respiration, all we mentioned was glucose, and it seems as though you would only need glucose to create energy in a cell. And that's not exactly true, right? This is a basic coverage of biology. We're not really diving super deep into it. However, this shows you where the carbs, fats, and proteins come into play in the process. You'll notice that fats are needed in the first 2 steps, proteins in the first 3 steps, etc. So don't assume that just because we didn't mention it, all the different macronutrients are necessary at some point in this process. The other interesting thing is if you look at this and then you flip it over, right, we can reverse some of these processes to create some molecules. So we reverse the citric acid cycle, we can add some amino groups and make some proteins. We reverse the glycolysis, we can create some sugars. Not we, but some organisms can. So again, we're going to drive biosynthesis of molecules. Once we break it down to create ATP, we're going to use that ATP to fuel the creation of molecules. Everything's a cycle. All right, so that's the end of chapter 6 on cellular respiration. Again, go through the videos, add to your learning, right? Ask me any questions if this is a difficult concept. So make sure you're asking the questions